Good morning, everyone. Today we will discuss a small topic and on bacteriocins. Okay. So we'll be talking about bacteriocins, followed by my second lecture will be antibiotics. So this is a very small topic we will take. Just this lecture number 10. The lecture is on bacteriocin. This lecture is intended again for the technologist UG and students as well as text. So this lecture will be mainly focusing on the bacteriocin. The outline of the lecture will be bacteriocin. What is it? What is bacteriocin? What is the difference between bacteriocin and antibiotic? How is it different from antibiotic? The general characteristics of a bacteriocin. Some of the cultures we will discuss, culture or you can say microorganisms which are used for bacteriocin, and what is the application. So we will be dealing with this four, okay, and the structure of bacteriocin. So in this lecture, I will be taking about bacteriocin. The outline of the lecture goes what is bacteriocin, the difference of bacteriocin and antibiotic, general characteristics of bacteriocin, the culture of the microorganism used for bacteriocin production, application, and what is the structure of them. So I'll quickly take this and my next lecture followed by this will be your antibiotics, so that it will be clear that how does antibiotic works and how is the bacteriocin, how is it effective? Bacteriocins are effective. So first I'll deal with bacteriocin. My first point is, what are the materials or what are the substances which is having some bacterial property, antibacterial property or antimicrobial property? They are biopreservatives. Okay, and bacteriocins will be mainly dealing with the protein-like molecules having some antimicrobial activity, and they have amino acid residue. So bacteriocins are having antimicrobial activity. So my first point is, what is bacteriocin? Like, so first, antimicrobial activity they possess. They have amino acid residues. Okay, and they are very much different from that of they are obviously different from antibiotics, and how they are different, that we will be discussing in our second point or second subtopic of this lecture. So here it is like having mainly the structure. If we are talking about, then definitely the structure will come bio. So bacteriocin is also called as a biopreservative also, and one such example is niacin. Okay, so bacteriocin, one very common example is niacin. So one common example is niacin. Other examples also we will just successively come to. Now, my second point is, what is the difference between a bacteriocin and an antibiotic? First thing is, when we are talking about bacteriocin and antibiotic, the first thing, we are getting the bacteriocin from a food grade bacteria. It will be safe. It is generally safe and food grade bacteria. I will omit the entire screen and I will come to this, so you will be able to follow it clearly. So first I will be talking about bacteriocin. I said what is it and what is the difference between bacteriocin and antibiotic. Bacteriocin is mainly produced by food grade bacteria. Okay. Antibiotic is usually a secondary metabolite in the fermentation. If you talk in fermentation aspects, it's a secondary metabolite and it is produced by non-food grade. Okay. Non-food grade microorganisms. The second point would be ribosomal synthesized protein. For this, it is ribosomal synthesized proteins undergoing post-transitional. It will be having some translational modification. You are aware of that translation, transcription. So it will go for some translation modification. For antibiotic, it is non-ribosomally synthesized and catalyzed by multi-enzyme complexes. So it is non-synthesized opioid and they will be mainly catalyzed by the enzymes. Catalyzed by enzymes. Okay. The third point is it is basically a large peptide. Usually they will have around 34 or more amino acids. Whereas if you talk about the antibiotics, they are non-proteins. They are small and actually it is smaller. Small peptides, we can get it synthetically, we can form it in synthetic in nature, like that. Okay. So if it is a small peptide, then what happens in the antibiotic? It is small. That means by what? They have smaller area if it is small. So diffusivity and surface area, differential permeability, these roles will come into play, isn't it? Small area, so it will be diffuse. It can diffuse, and differential permeability. These all will come into factor. That's why antibiotic will be more effective. Usually it is right. Then we have the next point is it is not affected by any enzyme. The antibiotics are not affected by enzymes in GI. It is not affected by enzymes in the GI tract. It is affected. No adverse effect. Actually, they will get destroyed. The fourth part, they will destroy by proteolytic enzyme. Okay, GI tract में proteolytic enzyme it will be getting destroyed. It will not be affected. But at the same time, if you say the fifth point will be like there is no adverse effect. Bacteriocin will not have any adverse effect. But there are chances of the penicillin to have adverse effect. Okay, and they will bind to for this bacteriocin. The point number 6, I'll write it here. For bacteriocin, it binds to specific receptors. Bind to specific receptors. Okay, causing viability loss. So it will bind to specific receptors. Viability loss is there for that specific bacteria. Whereas, if I talk about the antibiotics, they inhibit cellular function, not through binding. They will not bind. Okay, if they are not binding, then the viability will not be affected. So they are not getting binded, so its viability not getting affected. Okay, so if I say here what are the things happening, it is bacteriocin is a food grade, antibiotic is a non-food grade, ribosomally synthesized, antimicrobially is not ribosomally synthesized. Then the larger peptide as per structure, it is a small peptide, so it will have more space, surface area to get diffused through and it will be okay. Then the GI tract will not be getting affected. No adverse effect will come from that bacteriocin, but antibiotics definitely will have then upset, there will be some effects. Like upset of the GI tract. Sometimes when we pop the antibiotics, we are not feeling good. We are going for washroom. Okay, like that. Some stomach blockage, some stomach ache, like that. GI tract will be. Some cases it gets adverse impact. Along with that, we will have they will bind, the bacteriocin binds to the specific reactors on the cell wall barrier, and what they do is the viability of the cell gets lost. Whereas for that antibiotic, they will be functioning through bonding. There is no such binding to it. It will not bind. Okay. Rather, what happens? They directly through cell wall. They will directly get in the cell wall and penetrate through and attack the DNA. Okay, here the basic mechanism of bacteriocin is binding the specific receptors. But that antibiotics will directly penetrate through the cell wall due to the smaller in size, as I mentioned. Small size diffuses, penetrate to the cell wall, go and hit the DNA and kill that is how they will destroy the cell and effectively they will manage. Okay, this is the difference between bacteriocin and antibiotics. The next one we will talk about is what are the different, like few different types of biopreservatives we are having. And then after that, whatever the biopreservatives we have, then after that, we will be talking about the yes. So we will talk about biopreservatives. First thing, that is about the bacteriocins. What are the types? Okay. So the types we will be discussing here is about 2 or 3 around we will discuss how it is. Like the first and foremost is your nisin. Okay. The second one you can talk about Bacteriocin. These are the few names of bacteriocins which we can do, and it is Bacteriocin. And the other one is lactatin. Okay. So these are the types of bacteriocin. Okay. So the nisin is having around 34 amino acids structurally. And what exactly happens is they are having the antimicrobial spectrum is gram-positive bacteria they will work on against effective against gram-positive bacteria. Okay, and the microorganism is Lactococcus lactis. Mainly it is Lactococcus lactis. And that other, in my lecture 9, we have discussed some questions, right? Over there I have said Lactobacillus and Lactococcus is the main one. You may omit the Lactobacillus in that case. Lactococcus lactis will be the main organism. There I have mentioned two. Okay. Next we will be talking about Diplococci. Diplococci is basically it is Lactococcus. Diplococci we can get it through Lactococcus lactis or cremoids. And this is antimicrobial spectrum is Lactococcus lactis and cremoids only. That is what it is effective. Okay. Then the Videocina is like Lactobacillus plantarum. It is against Lactobacillus plantarum. Videocina is mainly from Pediococcus pentaceus. Then Lactacin B, which is like Lactobacillus acidophilus. Lactobacillus acidophilus. producing organism. It is antimicrobial against Lactobacillus vulgaricus and Helveticus. Okay. So here, two three things can be taken. That is, one is Lactococcus lactis is for Nisin. There are 34 amino acids, and then another one is Diplocockin is basically the Lactococcus lactis. It is effective against Lactococcus pneumonitis. Pidiosina is the name itself suggests Pediococcus pentaceus. And then it is Lactobacillus acidophilus, and it is against, mainly it is against the Lactobacillus helveticus and health. Okay. So this is what it is about. Next, right? What is about the next we will be dealing with the basic characteristics of, like you can say, where all the basic characteristics of this bacteriocin. Number one is, as we say, it is a grass that is generally recorded safe, food grade. Then it is hydrophobic, bactericidal proteins, effective against gram-positive bacteria. Then it is resistant to high heat and pH. High heat and pH. So these are some of the general characteristics, and some of the disadvantages we have, like not effective against gram negative. Okay, gram negative, it is not effective. Sensitive strains can go resistant. If the strain is sensitive, it may be, after some time, it may be a resistant one. Strain will be resistant. Then lipids will reduce the effectiveness. If the lipids are present there, it usually reduces the effectiveness of the bacteriocins. And the fourth one is, you can easily destroy by proteolytic enzymes. Okay, so mainly it can be destroyed by the proteolytic enzyme, and lipids, if it is there, it will reduce the effectiveness. It is not effective for the gram negative, and if the strain is very sensitive, it will become resistant to that. Okay, so all these things are the disadvantages of it. Now, with this, we just conclude the bacteriocins. This is a very so I'll just summarize in the nutshell. That is, what is bacteriocin? What all we discussed? What is bacteriocin? The first point. What is the difference between bacteriocin and antibiotics? What are the different types of what are the different types of bacteriocins we have, like Nisin, Pidocin, Lactin B, okay, and Lipocockin? What are the microorganisms or producing organisms responsible for those four types? And on antimicrobial spectrum we discussed. Along with that, the general characteristics of bacteriocin we have talked about. So with this, we conclude this lecture here. The lecture has been sessioned by myself, Pailras Gupta, research scholar, Department of Agricultural and Food Engineering, IIT Kharagpur. Thank you so much.